Photobiomodulation, or red light therapy, is a process by which light is used to activate the cells in our body. This is a phenomenon that was first discovered by a Hungarian scientist in 1967 by the name of Endre Mester. This he discovered while working with lasers as a side effect of the laser therapy. As a result, red light therapy was originally called LLLT, or low-level laser therapy. Scientists soon realized that LEDs, or light-emitting diodes, can also produce the same effect. Red light, having the longest wavelength of the visible spectrum, penetrates skin more deeply than the other colors in the visible spectrum. Since then, red light and near-infrared light have become popular modalities for the treatment of a number of medical conditions. Red light and near-infrared light can repair and rejuvenate tissues, having a variety of beneficial effects. The packets of light called photons that are absorbed by molecules in the tissues known as chromophores. The process is similar to the way our eyes sense light rays to form the images that we see. In red light therapy, light is absorbed by three main chromophores as it crosses the skin. In the outer layer of the skin, or the epidermis, light is absorbed by melanin across a broad range of wavelengths, from 400 to 700, which constitutes the visible spectrum, and beyond. In the deeper portion of the skin, or the dermis, hemoglobin is the main chromophore, and this absorbs light in a broad spectrum with two distinct peaks at around 420 nanometers and 580 nanometers. Throughout the layers of the skin, water molecules are scattered, and these to absorb light across a broad range of wavelengths. The biological effects of light therapy come from the photomodulation of specific structures in the cell. One of the most important structures is the mitochondrion. The mitochondrion acts as the powerhouse of the cell. It is here that all the energy of the cell is produced for it to perform its functions. When the mitochondrion is stimulated, the cell wakes up. Its activity increases, and it produces new hormones and proteins and important molecules called cytokines. Cytokines can increase or decrease inflammation. They can also help with the maturation of stem cells, or they can attract other cells to the site of phototherapy. The mitochondrion absorbs light mainly through a molecule that is found in its membrane. This molecule is called the cytochrome C oxidase, which absorbs light in a range from 670 to 950 nanometers, with the maximum absorption taking place at 810 nanometers wavelength light. These wavelengths extend into the infrared range of electromagnetic radiation. The absorption of light by the cytochrome C oxidase releases nitric oxide from inside the molecule. This unleashes a flood of secondary messengers into the cell, such as calcium ions and reactive oxygen species and CAMP and ATP and transcription factors. These increase the activity of the cell and lead to a production of proteins and hormones and cytokines like we mentioned before. Because the mitochondria act as the major chromophores in the cell, tissues that are rich in mitochondria are more sensitive to red light therapy. Tissues such as brain and muscle and nerve that are rich in mitochondria are more sensitive and therefore require a lower dose of red light therapy to produce a given effect. In red light therapy, more is not always better. The response of tissue to light therapy is consistent with the phenomenon of hormesis, which states that a substance in a low dose may be helpful, but in higher doses it can be harmful. This means that there is an optimal red light dose for best results. Surpassing this dose may not only impede the process, but may actually cause harm. In the skin, one notable characteristic of red light therapy is the activation of fibroblasts. Fibroblasts are the cells that produce collagen in the skin. Collagen is the protein that provides the structure in the skin that gives it its texture. In humans, aging skin is characterized by a loss in collagen fibers. This causes the skin to sag and produce wrinkles. The stimulation of fibroblasts can reverse this process by increasing the amount of collagen in the skin. This can rejuvenate the skin and eliminate wrinkles. I hope you found this video informative. If you did, like and share the video with your friends and family. Thank you for watching. Until the next video, stay healthy and stay safe.